Hello, my dear students. Today we are going to talk or to start with genetics part 4. In this part, I'm going to take an example about case of dominancy in addition to talking about cases of codominancy and non-dominancy. In case of dominant allele, we are going to take into consideration a disease known as retinitis pigmentosa. In this disease, we have to see if the allele of the transmission or responsible for transmitting the disease is dominant or recessive. First of all, we have a very important given about the disease and about the characteristics, and in addition to the consequences that come after this disease. Retinitis pigmentosa is a hereditary disease. It is the main cause of visual impairment. The disease starts by affecting night vision and reducing the visual field. It is caused by progressive degeneration of rod cells, which are photoreceptor cells of retina containing the protein rhodopsin. What is important from this given? The importance of this given is knowing the type of the protein responsible for giving the function in this case, which is the night vision. And you have to take into consideration or to pay attention to something very important here. All the time, the given at the beginning of the exercise is important to link it or to make a link between it and the conclusion at the end of the exercise. To understand the origin of this disease, so this is our mission. We have to know what is the real origin of this disease. We study the structure of proteins encoded by different alleles of rhodopsin gene. As you know, each gene codes for a certain protein. The protein here is the rhodopsin. The rhodopsin genes consists of 1044 pairs of a nucleotide and codes for protein of 348 amino acids. So, in this document, or it represents a portion of nucleotides of the rhodopsin gene and for sure the amino acid sequences of the corresponding proteins in the individual for normal and the affected. All the time we have to take the normal as a very important parameter, because once we have to talk about the affected one directly, we have to continue with this individual. Without the normal, we can't know the origin, we can't study the fact behind this disease. For sure, we can't study the 1044 pairs. That's why we take an example from 391 till 408. So, and here are the codes for certain amino acids. Once you need to translate it and you find the sequence of amino acids. Number one, you have to pick out from the text the cause of retinitis pigmentosa. It's easy. Directly you go, it's a hereditary disease, and this is the which causes this disease. Compare the two nucleotide sequences. With comparison, you have to take the given only. So you are given that 391, the first one, so start from it. As usual, what do we do usually? We compare from 391 till 408. All the nucleotides are the same except, so you move till here. You find here is the difference. So you count it, you find the number, and you mention where GG are replaced by TT. So here at the level of the nucleotides and at the level of amino acids, for sure you can't compare without finding it. You know, you have to transcribe the DNA molecule into messenger RNA and studying it codon by codon to find the sequence of amino acids. Similarly, you will say that. So, all the amino acids or the sequence of amino acids is the same except this one which is replaced by the other. So, what is the origin of the disease? For sure, it's a mutation, it's a hereditary origin of the disease. So, here you have to explain how the modification and the nucleotide sequence of the allele lead to the appearance of the previously mentioned symptoms of retinitis pigmentosa. Okay, so when you have to talk about the modification, don't forget to compare a little again. So once there is an alteration, a modification at the level of the DNA molecule, directly it causes a modification at the level of the messenger RNA. And in case of retinitis pigmentosa, the messenger RNA here it codes for a different amino acid. This different amino acid gives a different protein because directly the protein changes its structure, changes its 3D configuration, and once it is altered as a protein, it becomes a non functional protein. When it becomes a non functional protein, directly the rhodopsin cells will be affected and the vision will be affected, and this individual can't see at night. So, what do we have to tell you here? Don't forget to make a link between the result of the mutation and the given that you have at the beginning of the exercise by giving you the characteristics of the disease. In document 2, which represents the pedigree of a family having some of its members affected with retinitis pigmentosa, we have to determine in part 4 the genotypes of individuals 2, 3, and 2, 4. Before the determination, let's check if we can know from this pedigree if the disease is dominant or recessive. Individual 1 1 and 1 2 are affected. They have the disease. And you will see here they have children number 2 2 and 2 3 that are normal. Directly, what can we say? What does this mean? This means that the allele of normal is masked in these two individuals that are 1 1 and 1 2. So, on the other hand, we have to say that these two individuals are heterozygous. So, they carry the allele of the disease, but in the same time, they are also carrying the normal allele. But the normal is masked in 1 1 and it's masked in 1 2, which means that it is recessive for the normal and dominant for the disease because in one copy it expresses the phenotype. So, what is the genotype of 2 3? 2 3 is normal, recessive, and recessive can express the phenotype only in homozygous state. So, 2 3 is pure for sure, and the genotype is small n. What about 2 4? 2 4 is affected, so automatically, directly, he carries the disease allele, which is capital D, in order to express the phenotype. What about the second allele? The second allele can't be known if you take the parents of this individual, but directly can be recognized if you took the offsprings. So 3 4, for example, is a pure normal, and the 3 4 has to take the normal allele from 2 4 and a normal allele from 2 5. So in an obligatory way, he's going to inherit the normal allele from 2 4, and that's why there is only one option for this individual to be heterozygous, to be hybrid, and of genotype capital D, small n. If you took these symbols, for sure you can symbolize in your own way, but the disease is capital in any way. In part 4, 5, you have to read here woman 3 2 is going to marry her cousin 3 3, and we have to determine the risk of this couple to have children with retinitis pigmentosa. So easily we can go directly to Punnett square by finding the real genotype of 3 2 and real genotype of 3 3. Both of them, they have the same genotype. Why? First of all, they are affected, so they carry the D allele, and each is coming from a normal father here. So obligatory he's going or she's going to inherit the N allele. So this female is hybrid DN. This male similarly is hybrid DN. Okay, so Punnett square DN, what are the crosses? Crosses or the possibilities. So D allele with D allele affected, D allele with N allele affected, N allele with D allele also affected, N allele normal. So this couple for sure is in risk of having an affected child, and the probability is high here. It is 3 over 4. Why again 3 over 4? Because DD, DN, and DN, these three possibilities are going to give the disease. So it's a 3 over 4. And the risk to be, or it's not a risk, or the probability to be normal a child, it's only NN and it's 1 over 4. So by following the Punnett square, easily you can find the risk of having an affected a child. Here is the answer. So for sure you can read it very well, and you can study part 4, by the way, part 3 in a very important way. So in order to know what will be the consequences after any mutation. Okay, another topic I have to talk about it today is also cases of codominancy or non-dominancy or incomplete dominancy. What do I mean by this? Just look for this figure and you will see that. So, if we are taking complete dominance, what do we mean by this? So, this is blue, this is yellow. Once an individual carry both the alleles, he is blue. So what does this mean? This means that blue is dominant over the yellow, which is recessive. But in this case, a blue allele with yellow allele, an individual is blue spotted by yellow. Both colors appear. When both colors appear, we have to say that it is the case of codominancy. Both phenotypes appeared here. However, in this case, so when the blue allele is carried and the yellow allele is carried in the same time, and the genotype is CBCY, it is a new phenotype. It is a third new phenotype. This third new phenotype is intermediate between the blue and the yellow. So it is the case of incomplete dominancy or non-dominancy. Here, a chart which shows you the incomplete dominance or the non-dominance. As you see here, a pure red RR and a pure white, we put it small r, small r. So pink appears. When pink appears, it's a new third phenotype intermediate between red and white, and that's why we say that it's a incomplete dominance. Here we can change for sure the symbols and we can write it capital W, it's the same. Here we have to take the hybrid case. If the genotype is CRCW, CRCW, let's see the combination. When R combines with R, it's a pure red. W with W, it's a pure white. But when R is with the W, it's pink. Pink is intermediate between both of them. So let us find the proportions. So among the four, it's 1 over 4, 1 over 4, 1 over 4, 1 over 4. So for phenotypes, 1 over 4 red, 1 over 2 pink, 1 over 4 white. As genotype, 1 over 4 CRCR, 1 over 2 CRCW, and 1 over 4 CWCW. So as a ratio, there is 1 red according to 2 pink and 1 white. So it's 1 to 2 to white. The last example is about the case of codominancy and as an example, blood groups. So, you know that there are several blood groups. It could be A, it could be B, it could be O or AB. What do we mean by this? When it's A, we are talking about certain antigens, A antigens on red blood cells. B antigens on red blood cells, it's B. So no antigen, it's O. When A antigens together are found with B antigens, so the blood group is AB. Both are expressed, the A antigen and the B antigen. In this case, we can't say it's an incomplete. It's not an intermediate between A and B. No, together they are expressed, so it's the case of codominancy. Thanks for today.